Are you audible? Please start. Sir, are you audible?
Hello, you want me to start?
Yes, sir, please. We are eagerly waiting.
All right, can you hear me clearly?
Yes, sir.
There's a very bad echo that I can hear, so I don't know if my voice is coming through to you.
Yes, sir, it is coming.
I think everybody should turn off their phones, and then that might help give us a clearer sound.
Yes, sir.
Is that better? For me, I hear more clearly my own voice, but there's still an echo, which probably means somebody still has their phone connected. I know you have very important things, messages that you can get, but I hope you can close your phone for the next 30 or 40 minutes. Should I start now?
Yes, sir. Yes, sir. Start. Please start.
Hello?
Yes, sir. Yes, sir.
Continue,
Yes, sir.
sir.
All right, I can start. I think uh, A has said start, but I'm still getting a lot of background noise. All right, good afternoon. I am talking to you from Finland. We have about -4 degrees Celsius and a little bit of a snowstorm. That's quite warm because in the past days it has been cold, about 25 degrees minus. There's still people coming in, so I'll wait a minute. Now it seems that everybody has arrived. So I wish to start a very good afternoon to everybody. I have, of course, a very big difficulty. While I know something about West Bengal and about your city, I, of course, do not know you personally, and that puts me at a very big disadvantage as I try to talk something that would be of interest to you. So I've decided to do the following thing. I will take 10 important areas of my life and tell you a little bit about why open sharing and borderless collaboration is so important. In 1968, I joined a German company called Volkswagen and their office in Canada. My first job was to help the company understand what it meant to be in the age of the computer. You have to remember this was 1968, and computers took up the size of your room and were very, very awesome. People were very much afraid. And so I was asked to develop a program to show what the computer was and how it worked and why you should not be afraid of it. To do that, I had to get information from countries that had much more experience with computers than Canada had. And this was a very interesting experience to try and find out how they had overcome the difficulties. And I found out that many, many companies, many universities had not really succeeded in doing this very well. And yet I started work, and after a short while, I found out that we had no problem with our staff. And the reason that we had no problem is that they were very impressed, very, I would say, satisfied that the management of the company had taken care of their needs, taken care of their peers, and hired somebody like me to help them and to help the company through this dilemma. That was a very, very big step, and I learned very much about international communication by talking with people around the world about this problem. The second point I'd like to bring up in 10 years later, in 78, I was hired by a Volkswagen in Brazil. The Volkswagen company had more than 40,000 employees, which was a very large number of people in one company. And it was the largest car making company in Brazil. Uh, Again, a German company that was learning how to operate in Brazil. And Brazil had a very interesting thing. There were a lot of people, and they were very cheap. And so the companies were very content to hire people for a year or two, fire them before they had to pay any kind of indemnity or pension, and then hire new people. And this was very much the standard practice of all the companies in Brazil, especially the foreign companies. Our company was different. We decided that people were very important and they would invest in our company if we would invest in them. And so we developed a plan to provide individualized education for each and every one of the employees. To do that, we shopped around the world to find out what was happening in other countries. And the first place we looked at was Japan. and we saw how Japan used video equipment to provide very good on-the-spot training for their employees. We also looked at in Italy, and we found out that they had developed uh, certain games that at that time were very, very advanced simulations that again helped the employee understand what was happening and how they should fit into the systems, how they could learn, how they could get better at their job. So again, this was a case of taking the international experience that was much more than we had in Brazil, and bringing that to Brazil to allow the people to benefit from the experience of other countries. My next example is also from Brazil because some years later we decided to build a car in Brazil for the US and European markets. And this was a very, very bold step because it had never been done before. A third world country like Brazil producing a car for a first world country like the US or Europe. And for that, again, we had to get opinions of all the people that were involved, the workers, the users, the purchasing people, all the people that had a role to play in this development, and learn from them, from and in their countries, what this meant to them, what their problems could be, and how they think they could be addressed. And again, it was a very basic program of helping people understand the needs they had for training and development, and then how they could best get it. So this was a terribly involved process, but one that turned out to be very successful. And the new car was built in Brazil to meet the standards uh, of the countries, the legal standards, but more importantly, to meet the needs of the consumers in those countries. The next thing happened in the year 2000. And in the year 2000, I joined the United Nations in the Office of the Secretary General of the United Nations. And my task was to help implement something called the United Nations Global Compact. The Global Compact was Kofi Annan's initiative to ensure that companies and the UN spoke the same language. And this is something that is very difficult because even though people could speak English or French or Hindi, they had very different concepts of what things are. And it was our task to build a common understanding. And this required also, first of all, an understanding of the people. And that's not so easy. Uh, we tend to see things not as they are, but as we are. We see things through our perspective, through our eyes, through our experience, through our knowledge, and not so much from what the reality is. And it's a very big, big step for each of us to understand that the people we're speaking to in another country, very often the difference between men and women also creates barriers. Find ways to overcome these barriers and to communicate successfully between people of different cultures with different languages. And I think my experience here is that the difference in cultures is much more important than the difference in languages. Languages are important. We must have a common tongue, but that we can find ways to get around. Culture is very difficult because we do not have a common culture. And so that was a major learning step for me to overcome this barrier, to find ways to help people communicate with each other and to understand the importance of communicating, understand the importance of not just going to assumptions, saying, well, I would do it this way because the person in another culture would probably say, I'll do it in a completely different way. So that was very, very important. Uh, the next time I will go is 2004. In 2004, I was a professor in a university in Austria, and I went to a meeting in Vienna, the capital of Austria, a UN meeting because there's a major UN implementation center in Vienna. And I walked into the meeting and I sat down and I started to listen. I came a little bit late, and I found that the person that was speaking something that was very dear to my heart. He was talking about how one could improve quality of rice so that it contained all of the nutrients, especially the nutrients that women require if they will have a baby, and the baby will require that the mother is stocked up on these nutrients, micronutrients. And the man continued to talk, and I thought this is really, really interesting because I was very interested in hunger. I was very interested in malnutrition, and I'd been teaching some things about that at the university in Austria. It turned out that I was in the wrong meeting. The meeting I was supposed to be in was two doors over, but I decided I had invested so much time and the topic was so interesting, I would continue to stay in the meeting. At the end, I had the opportunity for a long talk with the speaker, who turned out to be the chairman of a very small genomics company, or actually a research institute in China. And the discussion was quite fascinating because I had never really known much about genomics and the importance of genomics in human development, and, and especially how it affects the lives of each of us. And so I stayed and we talked and we talked and we talked. And finally, I decided this is really interesting for me. And from that time on, I started to work with that company. The company at the time, which was an institute, a not for profit institute, had 46 people, all very highly skilled scientists. And like you are today, they were also at the beginning of their career, although they all had PhDs from countries around the world. And uh, we started to talk, and we started to see what we could do to help humanity be better. Some of the people in this group of 46 had been members of the Human Genome Project, which took place at the turn of the century and was one of the fundamental building blocks for genomics and healthcare today. Uh, we had a very long discussion how we could grow, where the problems were. And today, this small group of not for profit leaders uh, has about 10,000 scientists spread all over the world, including in India. And I had the opportunity to work with, while I was at the UN, with some of the leaders like the Ambani, the Tatas, family, the major leaders in India, trying to find out how they could work together with Kofi Annan to make the world better for business, to share in the development of a better life for all. And really, the Indian CEOs and the Indian government officials were very, very interested and collaborative and helped very much at the beginning of the Global Compact. And this really added a level of information, of knowledge, and of wisdom to what we wanted to do by adding the knowledge and the wisdom of India into the compact, into the, I think, thought processes of the global compact. And these times for me were very important because as a young person, I had always thought I would love to live and work in India. And this gave me a perfect opportunity to understand a little bit more about the Indian culture, to work with the leaders. And so I also met a very interesting person, Dr. Patak. Dr. Patak created something called SULAB. And Dr. Patak was very interesting because he, as a young PhD, a young scientist, was commissioned to do a study on the Dalits, on the untouchables. And after 2 months, he decided the last thing that these people needed was really another study. But he decided what they needed to do is find ways to change their employment, find new ways of working. And he created one of the world's greatest uh, toilet experiments, which today India, across India, benefits from his work. He was based in Beijing, in Delhi, and he really helped develop an international toilet uh, consciousness in India, with small toilet, medium-sized toilet, and gigantic toilets to help different parts of society have access to hygiene, which was very important, and that the hygiene would allow the development of valuable things like grasses and gas that could be used as fertilizers and for heating and cooking and lighting. So this was, again, an international approach because he had to go around the world to find experts who had really this kind of experience and were willing to share with it. And his project was a tremendous success and is still a great success in India. So this again is another example of how open sharing can help. And it's very difficult if you keep hiding things or if you keep only giving part of the information. It works much better if you give full information and you define your problems very, very completely and very, very succinctly so that people can understand and take on different things. To do this at AGI, at this little company, which is now a giant NGO, a giant institute, we developed something called the International Conference on Genomics. And every year, or sometimes twice a year, scientists from around the world, including from India, will join their fellow scientists uh, to understand the latest developments in science, in genomic sciences in particular. And today, for instance, the impact of AI on genomic sciences, which is tremendous, but it's tremendously beneficial if you understand how it can be best used. So this ICT is a very important link, and we create various networks. I work one network with a number of Indian scientists where we are very interested cancer development, cancer diagnosis, and cancer therapy. And this is again, very important to get the details of what works in different cultures and what doesn't work. And then to work together using this combined borderless collaboration to develop approaches, to develop instruments, to develop algorithms that really help, develop training systems for the doctors, for the diagnosticians, for the nurses, for the public health workers. All these people play a tremendous role. And again, this cannot be done without this kind of international collaboration. Today, I'm working on another project, which is called the BRI, the Belt and Road Initiative, of which India is one of the members, to help develop approaches where the information and the knowledge and the tools can be shared very easily, especially in the least developed countries. India is no longer a least developed country, it's a highly developed country. The expectation is by the end of this decade, India will have become the third largest economy in the world, and shortly thereafter, it will probably overtake China. So India is a very advanced uh, country, and it needs also to share its knowledge. Every day I read papers from around the world, and every day the number of Indian papers written by top scientists come to my desk, and I have the great pleasure and honor of making sure that they reach the hands of scientists, and in this case, of cancer scientists around the world. And I think this is built again a certain amount of network and friendship that is very useful in terms of developing solutions to problems. We also are facing potential problems with pandemics, and they are sort of buzzing up in different corners of the world. Some are just at the beginning, some are already have become very dangerous. And again, we have to fight this situation with collaboration, with sharing of information. One of the problems with sharing information that the data files for genomic information are enormous. And every country has its rules and regulations if they can be shared outside of the country. In most cases, the country would rather not share this information. And a group of scientists have developed an interesting approach, which is called data visitation. In other words, the data will stay in the data files, the data bank of the country or of the individual hospital or organization. And the people who need the data, access to the data, can do it by visiting and the data that they want with the approval of the data owner. And in this way, there is really comprehensive sharing of data without any of the risks of data being misused or misplaced. It stays where it belongs. So that a little bit is what I wanted to share with you this morning, and I hope that this is of interest to you. Now comes the most important part, and that is the questions that you would like to ask. I will try my very best to answer them. Thank you very much.
Thank you, sir, for nice words. Now it's open for interaction with the audience.
I'm sorry, I
Audience.
don't hear what you're saying because there's such a very large echo. So I have no idea what you have just said.
For this kind of I think I'm
See, I think the echo is not in the room, but in the electronic system.
Thank you, sir. Thank you, sir, for such kind of nice presentation and your valuable time. Thank you so much, sir.
What I understood was thank you very much for the presentation. If that was what you said, then I could hear that. But I'm more interested
Any
in
questions from students?
getting your question.
Any questions? Any questions from students? Please.
Regarding open access,
This is open access. Please, any question?
any
Raise your
questions regarding open access?
Okay, I think I can provide a way to do this. My email address is Fred, FredDubee, Doobie at gmail.com. The conveners know my address. If you will send me your questions by email, uh, not necessarily today, but in the next days, I will try to answer them as quickly as possible.
Okay, sir.
Would that be okay?
Yeah, sir.
Okay.
Okay, sir.
Okay, if any queries, any questions, students will ask you via email. Okay, sir. Thank you, sir.
For me, your questions are so important, and that is the way that we can have open sharing. If I can understand what you would like to know, what you know a little bit about me from my experience. I have given a copy of my CV to your administration, so you can get some data of what I have done and where interests are. And from what you have been able to understand from this echo-filled presentation, you know a little bit the areas that interest me and that I've been working on for the last 50 or so years.
Yes, sir, we think our students they are enriched with your valuable lecture. So thank you, sir. We will say our student to contact us via email if they have any question regarding the open access. So
Okay, sir.
Okay.
Thank you once again
thank
for
you,
giving us
sir.
your valuable time. Thank you, sir. Have a good day.
Thank you, sir, for giving us your valuable time. Thank you so much, sir, for presenting here. And
Okay. Thank
it's
you.
such a
Thank
fine
you so
lecture.
much. And I'm just sad that we've had such a problem of communication, but this is not, of course, between countries uh, with different technologies. This is really uh, not an unusual situation, but we have to find ways to overcome it. Uh, it would be wonderful if we could have a second conference. I would be very happy to work, find a way that we can do it without echo.
Thank you, sir. Actually, there was some problem regarding the Zoom link. That's why we request you to join in this Google Meet link. There is some issues and network issues were there. So sorry for that. And sir, thank you very much. Although there are several complications, network issues, but your lecture enriched us so much. Thank you, sir. Thank you so much.
Okay, good. Thank you very much. Our time is up. The time I was given by the administrators, so we have to close now. But I'm very much looking.
Thank you, sir. We will
Thank
again.
you.
Thank you, sir.
Thank you, sir. Have a good day.
Have a good day, sir. Thank you.
Okay, bye bye.
Bye bye, sir. Again, we want to hear from you in future. Thank you.
Okay, welcome online presenters in the technical session 2. I'll be calling the names. Please, if you are present, please respond. Uh, OP6, Chittaranjan Dash, are you present?
Yes, I am present here.
Yeah, okay. Please mute.
I am present, ma'am.
Sir, please mute right now. Sir, please mute. I'll be calling your. Okay, sir. Go on with your presentation. Chittaranjan Das, he will be presenting on the titles, litter production, and decomposition dynamics of secondary seeds in Gadwan. Sir, can you hear me?
My slide is visible. It is visible.
Indrani Banerjee, please can
My
you mute?
slide is visible.
Rest of the presenters, please mute your computers. Attention, please. Rest of the presenters, please mute. Yeah. Chittaranjan Das, sir, please
My,
go on with your presentation. Yeah.
my slide is clearly visible. Yes or no?
Yes, sir, it's visible. The time period is 8 minutes for your presentation and 2 minutes for interaction. So please try to confine your presentation by that time. Okay, sir. Please go on.
Okay, my voice is clear.
Yes, sir.
My
Yes,
voice is
yes.
clear.
Yes, absolutely. Please go on.
Okay. Good afternoon to all. Today I present my paper, the International Conference on Next Gen G Bioresource Frontier for Sustainable World, organized by Science Department, Asansol Girls College. Today, my topic entitled litter production, decomposition, dynamics of selected tree species in a forest area of Bardhaman, West Bengal. Now, this what is litter production? Litter is the leaf shed there material, waste material or extreme material in a particular season or a year. Basically, during after the end of the winter or start of the summer period, these leaf litter are fall on the ground in a forested area. So these leaf litter are very essential for
Sorry to interrupt you, sir. Sir, the screen
uh,
that you are presenting, please can you close the small window because it is not the full screen is not we can't see the full screen of the presentation.
full not visible.
No, so there is a small window open of your login. If you can please remove that when you are
Okay, okay, I
presenting.
try. I leave and try again. Okay.
Sir, you minimize your login. So just you minimize it. Yes.
Okay.
Now it's visible. Yes, sir. Yeah, yeah. Thank you. Yeah.
Uh,
Go on, please.
okay. So leaf litter and decomposition is the key process, natural process in the any terrestrial ecosystem, specifically forest ecosystem, which enhance soil nutrient cycle and provides detritus food chains, food, and also remove waste, reduce soil fertility, and maintain carbon sequestration. And during decomposition, significant amount of carbon dioxide release. It is very warming in situation during decomposition, and also release some different form of ionic form, ammonium, nitrogen reaction, phosphate, etc. And decomposition is controlled by mainly biotic, abiotic factors, and abiotic factors, and also litter quality, soil property, and decomposers community, and any others numerous factor depend climatical or meteorological factors to depend on litter decomposition. And this also litter several quality factor like lignin, carbon, phosphorus, nitrogen, carbon nitrogen ratio, carbon phosphorus ratio, lignin nitrogen ratio, etc., cellulose, etc. So why interest with decomposition study? Because you can decomposition influence carbon cycle and local and global scale, release nutrient for plant uptake, and renew energy for microbial growth, and also biological process without decomposition is stopped. So decomposition is essential for natural process and change in atmospheric carbon dioxide level. So why litter is important? Leaf litter is the most of the carbon nutrient transfer from plant to soil by the leaf litter only, and also some bark and branches also. So the leaf litter maintain moisture, temperature regulation in soil, provides habitat, added nutrient and the soil protection, maintain biodiversity with the forest ecosystem, provide continuous energy source, macro and microorganisms, and also provide food and shelter for arthropods and large insect and countless other animals. At biosphere level, decomposition is very important because significant amount carbon dioxide, methane, nitrogen gases released. These are also called greenhouse gases. So very interesting decomposition. And soil is the major source of carbon. So decomposition study is very essential. And what control decomposition? Decomposition is controlled by mainly litter quality, climatic factor, meteorological like temperature, rainfall, humidity, and biotic factors, and also soil quality, soil fauna, and soil microbes. So several literature study abroad all over the world study litter fall, litter decomposition, nutrient release, and nutrient transfer to the soil from plant to soil. This is the several literature has studied. Now, what are the objectives? First, estimation of litter fall collection by litter traffic. Understand the pattern of litter bag technique, determine the nutrient level of leaf litter for different species, and to understand major nutrient release from litter, and to understand degradation influenced by litter quality and soil uh, quality. This is a study side by Bardhaman, this India, also India, West Bengal, Bardhaman district, mainly for Bardhaman, and these are Ramya Wildlife Sanctuary study site for litter collection and litter field study. Now, this methodology, first of all, study framework. This selected site of the study site was, and then collection of litter fall, natural litter fall by the litter trap. This is the litter trap, nylon trap methods, nylon like mesh type method, box shape framed and this placed on the tree site. And after collection litter, then dry and wet and put in a litter bag. This is a nylon mesh type bag in a specific amount and for study decomposition in the field condition. After several month intervals, I collect the litter bag and measure this weight, initial weight and final weight. They study decomposition rate and also study nutrient analysis after collection of litter bag in a certain time. Then dust or mixed litters and dust, and this dust sample analyzes several chemical parameters on the litter. After decomposition of litter, this looks like only 10. So any organic material sustained disappeared after decomposition study. Now this is the overall study framework for litter collection, preparation, feedback and field study, decomposition study, chemical analysis, measurement of nutrient release and decay constant and decay parameters also half life, etc. This is the principal materials. I choose only 4 selected species: Albizia libec, Shoria robusta, Technogandis, Cresera oleosa. This specific species is used because these most of the species are deciduous species. So shed their leaf in a particular season in a year. So large amount of leaf fall and nutrient transfer these plant from plant to the soil. And this is the chemical method analysis by leaf litter, and this is a soil parameter analysis standard method. Now, this is the litter trap place, the center, this specific distance from one circle to another circle 5 meter apart, and alternatively place the litter trap for collection leaf litter collection, and this is the turnover rate calculation by this formula. Now, this is the place of litter bag on the forest floor. This specific pattern, randomized block, is deployment of the litter bag. Now, this litter bag after time 1 month, 2 month, 3 month, etc., collected the litter bag and the laboratory and dry, weight, and then final weight and calculation decay constant by the formula. This decay constant by the standard formula and
Sir,
also
one more
synthetic
minute
force
left. Please try to wrap up your presentation by that time.
and also 50% decay, 95% decay also calculation and also residence time, RT time calculation, and relative decomposition rate also calculated by this formula, standard formula. Now this discussion. This is the forest data of specific 4 plant leaf litter collection total million hectare per year. This Surya Robusta highest, and then Ectonagon is oleosa is living. And above ground biomass also calculated by this formula. I already mentioned the above total biomass calculations by standard formula, Biometal 1989, and then age, height, waist height, basal area, volume, etc., calculated by standard methods. Now, litter fall collection are mainly regulated by temperature. Generally, low temperature period, maximum litter fall occur. Generally, winter season, generally deciduous forest shed their leaves. This study graphically represented by calculation by figure the temperature and litter fall in 4 pieces. Now also rainfall pattern or precipitation also
Sir, please
related with
conclude,
the
sir.
litter fall. Generally, high rainfall period litter fall collection are lower. A lower litter for rainfall period, higher litter fall collection. And this is rainy variations, rainy, summer, winter seasons. Now, temporal variation liter nutrient, leaf liter, fallen liter, winter liter, rainfall liter, rainy period liter, and senescence liter. So, variation their nutrient concentration, specifically I studied three nutrient, nitrogen, phosphorus, and potassium, which is essential for plant for nutrition or renewal soil nutrient. And this chemical several parameter also studied nitrogen, cellulose, lignin, phenol, water soluble compound, lignin by nitrogen ratio, carbon by nitrogen ratio, nitrogen by phosphorus ratio, phosphorus, carbon by phosphorus ratio. So different type or ratio of chemical component also analysis by four type of liters. Now, decomposition also predicted by previous model. First is this graphically presented. Initially, little
Sir, please
lag
conclude,
phase.
sir.
Hello.
Sir, the time is over for you. Please try to conclude as fast as possible.
Time time period কত?
8 মিনিট ছিল স্যার।
ওকে, সেটা জানা ছিল না আমার।
আমি বলে
ঠিক
দিচ্ছি
আছে।
স্যার।
Now, type, ঠিক আছে, completely type. This decay exponential model component by several single exponential model, also linear regression model, also apply multiple regression model would fit this model by 90 more over 0.9% percentage radical value, and temporal variation of decomposition liter fall in different period also shown, and percent correlation related with mass loss, specifically lignin, nitrogen, and carbon nitrogen ratio negatively correlated, but phosphorus, potassium, and water soluble component positively correlated. Also, several parameters also correlate with the liter decomposition ratio, specifically organic matter, nitrogen, and phosphorus. And this single exponential model also apply carbon, nitrogen, phosphorus, and potassium. So decay constant, 50% decay, 95% decay, and the Asper values. And this principal component analysis, so this circle means could be positively correlate less carbon ratio in Surya Robusta, and less carbon nitrogen.
Sir, there are
Uh,
many presenters, sir. Please conclude. You
okay,
have
okay.
already
This
exceeded your
nutrient.
time, sir.
Okay, within 1 to 2 minutes, try to complete.
Now, 1 to 2 minutes.
Several.
You have already
Hello.
taken more than 10 minutes, sir. So please
Okay,
conclude. Conclude now.
ठीक है,
Yeah.
ठीक है।
Yeah. Conclude now.
Nitrogen, phosphorus, এবং potassium concentration will gradually change after several intervals of the decomposition study, and potassium mainly the fast release due to soluble leachable compound easily washed out from the liter, and also nutrient flux amount with the liter, nitrogen, phosphorus, potassium maximum, nitrogen, phosphorus, potassium also is Fourier Robusta. And this study of the same study liter, and after decomposition the several changes of the liters. Now, conclusion from the experimental study revealed that technogan is highest decomposition rate compared to other leaf liter, and initial liter quality lignin, nitrogen, carbon nitrogen ratio positively, and phosphorus, potash, and nitrogen positively correlated, and potassium is rapidly released to the leaf liter, so nutrient compared to neutral leaf liter. And further study to other microorganisms also responsible for decomposition. Any other plant is also not only for limited this, and this difference. Thank you.
Thank you, sir. Is there any question? No. We'll move on to the next presenter. Dr. Moinak Banerjee and Dr. Orhun Mondol. They'll be presenting on the topic recent trends and relevance of sports fisheries in psychological development. Are you there, sir? Who will be presenting? Dr. Moinak Banerjee or Dr. Orhun Mondol? Not present. Okay, the next presenter. Shampa Mondol, are you there? No. Indrani Banerjee Chakraborty.
Yes, ma'am, I'm here.
Yeah.
Can you hear me? Am I audible?
Yes, ma'am, you're audible. She'll be speaking on a topic, crop-specific modulation of nuclease activity in spinach and maize. Ma'am, please
Yes.
go on with your presentation.
Yes. Is my slide visible to you?
Yes, ma'am.
Yes. Uh, uh,
Ma'am,
Good
please
afternoon,
confine
everyone.
your presentation within 10 minutes, 8 minutes for your
Yes, yes.
presentation and 2 minutes for introduction. Okay, ma'am?
Yes,
Okay, thank you.
yes. Good afternoon, everyone. I am Indrani Banerjee Chakraborty, and today my topic is crop-specific modulation of urea's activity and soil microbial dynamics in spinach and maize, rhizosphere of the East Calcutta wetland, and role in nitrogen transformation. So, moving on to the outline of my total presentation. This is the small outline, and moving on to the next slide, which is the introduction slide. As we all know, urea is very much used in our agriculture for the plant growth and development, as there is a huge demand of food for now, but it is sometimes negligible that the activity of urea not only in our life and in environment, it is very much harmful for us. So it can degrade the soil quality, it can degrade the water quality, and so on. So basically here, this is the present scenario. And moving on to my next slide, this scenario is also applicable in the wetland ecosystem. I mean, this wetland ecosystem, in especially the Ramsar wetland ecosystem, which is very vulnerable to different types of stress condition. So our main focus in this study is to evaluate the effect of excessive use of urea in Ramsar wetland. Like here we choose the East Calcutta wetland, which is a popular wetland in West Bengal. So here, this is the main objective of my study, where we have investigated the physiochemical changes in the rhizospheric soil under conventional urea application in comparison with uncultivated and unfertilized control soil. And secondly, we have measured the structural composition of the rhizosphere microbial community, comparing the fertilizer plots with unfertilized and uncultivated salt. And next, we have also evaluated the ureolytic activity across conventional urea treated soil and control soil. And next, we also assess the antibiotic resistance profile of the rhizospheric bacteria isolated from the fertilized and unfertilized soil system. And lastly, we also examine some of the stress conditions like UV tolerance microbial population across cultivated fertilized and unfertilized soil. So basically, we have collected soil from the uncultivated and unfertilized soil from the East Calcutta wetland, and two soil samples were chosen from the from the corn rhizospheric soil of maize and rhizospheric soil of spinach. As these two plants are very much popular in nowadays, so we have evaluated the effect of excessive use of urea in this vulnerable ecosystem, and their potent, and we have also evaluated the alternative to this excessive use of urea. For the physiochemical analysis, we have analyzed the moisture content, water holding capacity, pH, electrical conductivity, total organic carbon, total calcium carbonate, bulk density, lime requirement. We have assessed these total 8 parameters in all the soil samples to evaluate their effect on the nitrogen stimulation and nitrogen use. And next, we have also evaluated the microbial enumeration and urea's activity, where we isolated the bacteria and fungal fungal microflora. And next, we also isolated some ureolytic bacteria, and we have also studied the antibiotic resistance pattern of this isolated microorganism. And lastly, we have also evaluated the microbial stress adaptation, like the UV tolerance. And moving on to the next slide, here we can observe that these physiochemical parameters and how these physiochemical parameters are altered with the uses of urea. Like the pH in the soil can be decreased in the spinach rhizospheric soil, and in electrical conductivity is also increased in spinach. But in the blank soil, which is uncultivated and unfertilized, there is as there is an presence of native bacteria or native environmental condition, there is no structural difference or there is no structural alteration in this type of soil. But when we analyzed the rhizospheric soil, it was observed that maybe for an instant the urea application may be beneficial for the crop production, but for a long run, it is detrimental for the plant, soil, and as well as the nearby environment. Next, there are some species which was isolated from the rhizospheric soil. In the spinach soil, total 6 isolates were there. In the blank soil, where there was no cultivation and no fertilizer use, we isolated 6 isolates, and in the maize rhizospheric soil, we isolated 2 bacterial species. These are some of the results of the bacteria and fungi which was isolated from the uncultivated and unfertilized soil, where we can see the fungal population and variation in the fungal population. Next, this is the these are the plates of the rhizospheric soil of spinach under conventional urea. Some bacterial isolates and some fungal isolate, and these are the isolates from the rhizospheric soil of maize. And here is a very important slide where we can see the abundance of ureolytic bacteria, where the media was supplemented with only 2% urea. And here we can observe that rhizospheric soil of maize and spinach, there is abundance of some red color or orange to red color pigmented bacteria. But in native soil or the soil which is uncultivated and unfertilized, there is a small abundance of ureolytic bacteria. And evident that this type of soil is more prone to have the ureolytic bacteria. Now, this is the antibiotic result, where we can observe that beta-lactamase type of antibiotic is more resistant to these isolates. And lastly, we have observed the tolerance level of UV according to the isolated species in spinach, maize, and uncultivated and unfertilized soil. And we evaluated how long these isolates can tolerate the UV. And lastly, we observed with the standard curve of urea that and mathematical deduction and using some mathematical formula, how much urea is left to the soil and how much urea is produced by this rhizospheric microflora and rhizospheric bacteria. And in the conclusion or discussion part, we can say that this whole urea application can disturb the soil physiochemical stability. Maybe it is useful for a short run, but it is not useful for a long run and sustainable agricultural practice. It can restructure the rhizospheric microbe, where we can see the crop-specific rhizospheric microbes alteration. It can accelerate the urea's activity and nitrogen turnover. It reduces the nitrogen use efficiency. And here we can also observe that antibiotic resistance is somewhere or somehow related to the application of urea. And in a whole, we can say that compromising long-term soil resilience and wetland ecosystem integrity. And it concluded this application of urea as a popular fertilizer is not useful for wetland ecosystem for a long run and sustainable environment. And these are some future aspect of my work where these isolates can be sequenced, and we can further analyze the resistance pattern, and we can also analyze the nitrogen loss qualification, and further we can also design a controlled release fertilizer, and we can have some specific program for wetland ecosystem sustainability. We can develop integrated nitrogen management program, maintain microbial functional equilibrium in Ramsar designated agro ecosystem. These are some references on which my work is based on. And thank you.
Thank you, ma'am, for sticking to the time. Any question? No. Any question? Okay, ma'am. We move on to the next presenter.
Yes, thank you, ma'am. Thank you.
Samjit Sabam Bharati Devi. Are you there?
Yes, ma'am, I'm here.
Yeah, she will be presenting on a topic estimation of tree biomass and carbon stocks in the Darjeeling Himalaya. Ma'am, please proceed with your presentation and please be with your time 8 minutes for your presentation and 2 minutes for introduction.
Ma'am, is my slide audible? Is visible?
Yes, ma'am.
Okay, ma'am. Okay. Good afternoon, everyone. Today, am I audible?
Yes, yes.
Okay. Today, I'm going to present on the topic estimation of tree biomass and carbon stocks in the Darjeeling Himalayas, India. And as everybody know, the Himalayan biodiversity is a Himalayan region is a biodiversity hotspot, and especially when we compare the two Himalayan side, the Eastern Himalayas and the Western Himalayas. Eastern Himalayas has high forest covered, and the protected area coverage is more profound, and it is reported to have high diversity of flora and fauna. And we also know that the Eastern Himalayas is threatened by the climate change and the land use pattern. And everybody is now aware with the concept that what is carbon sequestration? Carbon sequestration is the sequestration of the atmospheric carbon dioxide to labile and the recalcitrant pool. And when we talk about the carbon sequestration, especially the terrestrial carbon sequestration, to understand the tree diversity, estimation of the tree biomass plays an important role in carbon sequestration. And biomass carbon sequestration in the Darjeeling Himalayas is not well understood. So these are my objectives. First objective is to estimate the forest biomass of the forest, four forest stands, which is present in the three protected areas in Darjeeling Himalayas by using non-destructive method. And the second objective is to compare the values of above ground biomass, below ground biomass density, above ground biomass density, and the total carbon density in different forest types in Darjeeling Himalayas. And the forest types which was found in my study area are tropical, temperate, and subalpine forests. As you can see in this slide, this is my study area. I have conducted my studies in three protected areas, that is Singalila National Park, Nura Valley, and Mahananda. This work was conducted when Darjeeling Himalayas was not separated into Darjeeling district and Kalimpong district. The sampling was done in the monsoon, in the post-monsoon in the year 2020 and 25. I have done the sampling of the trees by using the standard sampling method, that is 20 meter into 20 meter for the trees. And the total area which is covered is around is 128 quadrates covering 5.12 hectares. And in the studies, I have specifically collected the diameter at the brace height, and the height was being measured. And this two data, that is the diameter at the brace height and the height, was fitted into species specific standard volumetric equations. And the biomass density, above ground biomass density, the below ground biomass density, then the total biomass density and the carbon density above ground and total carbon density was calculated by using the standard methods. And as you can see, the volumetric equations, I have used it from the Brown et al. And this is a sample study, a sample for all of you to see how the volumetric equation has been used. The site Brown's method I have used to calculate the wood density. This is the volumetric equation that I have used for different species in my study area. So I have also used biomass expansion factors for the different forest types and for the different trees. So this is the calculation in which I have indulged in my study, how to calculate the below ground biomass. So when we do the accumulation of the above ground biomass density and the below ground biomass density, we get the total biomass density. And so this is how I have calculated the above ground carbon density, the total carbon density. Then I have converted into total carbon percentage. So the result part which I want to focus is that the above ground biomass density was highest in the Mahananda Wildlife Sanctuary, which is the tropical forest, followed by the Singalila National Park. I have divided the Singalila National Park into Upper Singalila National Park and the Lower Singalila National Park. The Upper Singalila National Park has the alpine, and the Lower Singalila National Park has temperate, which I have grouped it with the Neora Valley National Park. So when we see that the lowest elevation, which is the Mahananda Wildlife Sanctuary, has the highest above ground carbon density as well as below ground carbon density, which is followed by the temperate forest, which is found in Singalila National Park Lower and the Neora Valley National Park. But the lowest above ground biomass and the below ground biomass and the total carbon is found in the Singalila National Park, which is predominantly dominated by the alpine forest. So this is a brief result which I have briefed. This is regarding the tree composition and diversity. As you can see in the slide also, the above ground biomass is highest in the Mahananda. And in the total carbon density also, you can see it is highest in the Mahananda Wildlife Sanctuary, which is predominated by the tropical forest. Uh, So I have also compared my study with other studies which has been done in the regional level, national level, as well as the international level. And the same pattern was seen, the above ground biomass, below ground biomass, and the total carbon density was seen in the tropical forest. So with this slide, I want to conclude. Thank you, everyone. Is
Thank you, ma'am. Any question? Thank you. Anyone present from OP7 and OP8? I had already called their names, they weren't present. Um, okay, the rest of the technical session will be carried on by Dr. Shuma Gorai. Thank you. Thank you so much. Thank you. Thank you, Shruvi. It is my honor to serve as the chair for this oral presentation segment. Before we begin, I would like to briefly outline the session guidelines. Each presenter will have 8 minutes for his deliberation, and it will be followed by 2 minutes question answer discussions. Okay? So, without further delay, we would like to invite our first participant, Mr. Srinjoy Ghosh. Are you there?
Yes, ma'am. I'm here.
Okay. Your deliberation topic is phytobeneficial biocontrolling bacteria in potato. Please proceed. OP
I am
24.
audible, ma'am.
Yes, ma'am. Yes, sir. 24.
My slide is visible, ma'am.
Yes, it is visible, but yes, minimize this.
My slide is visible, ma'am.
Yes, yes, it is visible. But a small
And I am
window is
audible, ma'am,
opening.
clearly.
Please close this window.
Yes.
Yes. Now it is okay. Please proceed.
Okay. So good afternoon to all respective dignitaries and teachers and students. Myself, Srinjoy Ghosh. I am PhD Research Scholar, Department of Botany, Coachbihar Panchanan Burma University, and Senior Research Fellow, Directorate of Research, Uttarbango Krishi Vishwavidyalaya. So today, my oral presentation topic is phytobeneficial biocontrolling bacteria for sustainable management of bacterial diseases in potato, under the guidance of Dr. Krishnendu Pramani and Dr. Nandita Sharma. So let's start my presentation. So introduction. So potato is the scientific name is Solanum tuberosum. It is a solanaceous crop, and it's in India ranked as the second in global potato producing. And also West Bengal is also in the second in total potato production in India. Potato is a starchy tuberous crop, and it is high in this vitamin C, vitamin B, potassium, phosphorus, iron, etc. But the bacterial diseases are the most important biotic constant in potato production. There are seven different type of bacterial diseases that severely damage in the tuber portion, which is the most economic portion of the part of this plant. The major bacterial pathogen and the disease name is bacterial wilt caused by Ralstonia solanacearum, as well as black leg soft rot caused by Pectobacterium and Dixia species, as well as common scab, which is caused by Streptomyces species. Then the current management practices of these bacterial diseases of potato is cultural practices. In these cultural practices, there is irrigation practices, field sanitization, crop rotation, etc. As well as the chemical control is copper-based using the copper-based bactericides, as well as use of antibiotics, etc. And this physical method, hot water treatment, and as well as the resistant variety, selecting the resistance cultivar for different bacterial diseases. This is the current management practices. But there is a little bit demerits of this current management practices. This is the limited effectiveness of bactericides, resistance in antibiotics, extensive practices, laborious, as well as the it is a non-sustainable practices, and there is a limitation of this availability of resistant variety. So in this aspect, there's a need of this biocontrol practices, which is reduces the chemical dependency, preserve the soil, water health, as well as the sustainable practices and real action, as well as disease control and this plant growth promotion. So this is the topic is the phytobeneficial biocontrolling bacteria. It's called as PBB. PBB is a class of plant growth promoting bacteria which is improve the plant growth as well as plant health. Plant phytobeneficial biocontrolling bacteria work with this type of specialized microorganism work in a dual mechanism. It is protect against diseases, improve nutrient availability, stimulate growth, and improve the stress tolerance. So next, this is the topic of this review. And last 18 years, we see that against this Alstonia solanacearum, bacterial wind causing pathogen, there is a 37% of this bacillus strain is reported against this bacterial wilted and brown rot caused by Alstonia solanacearum, as well as 37% of Pseudomonas strain is reported. Other than there is an Enterobacter, Serratia, Rhizobium, this type of strain is reported. See that in the bacillus, there is a detailed report of this. See that there is a 80.99% biocontrol efficacy, so that bacillus cereus strain reported by this Selimetol, as well as this bacillus subtilis strain, they got 79.37%, as well as another bacillus subtilis strain 82.7%. Another bacillus amyloleucopenicillin strain, that's the bioorganic fertilizer, uses 92% biocontrol efficacy in field conditions. There is a mechanism that is reported that when we use this phytobeneficial bacteria against this Blastoria solanacearum, they produce in this PBB by seed siderospore protease, chitinase, ammonia, IAS type of compound, and this is in this plant that they induce resistance by increasing total phenolic salicylic acid, peroxidase, polyphenol oxidase, phenylammonium, ammonia's lyase, and decreasing the catalyst. But there is a lack of this molecular mechanism against this antagonistic activity of this PBB against this diastonia solaniferum. Then we come from the another disease, another major disease of this common scab caused by streptomyces species. In these cases, we find that 50% of this bacillus that reported as a PBB against this streptomyces species. And this type of we see that there is a 38.2% of this drone inhibition in vitro condition and 82.50% of biocontrol efficacy. We see that there is a bacillus altitudinous strain as well as there is again is the bacillus amylolymphocytin strain reported by Lean et al. That it decreases the disease severity from 55.6% to 4.2%. And the mechanism is that against this pathogen, this streptomyces species, we see that they down-regulated that one gene, that is taxomin A biosynthesis gene in the streptomyces species, and upregulated certain type of genes in this potato plant system, that is chitinase, lacase, this type of gene. And production by this PBB is an antibacterial compound, is azalomycin, isopropanol C, that type of, and this lipopeptide is produced as well as surfactant, ethurine, fengicide, as well as if some certain plant growth promoting trait also produced, that is siderospore, phosphate solubility activity, etc. There is a certain type of compound which is produced by the PBB as a antibacterial compound. There is a phenylalanine A, azalomycin, bacillicin, isotropolone C, fengicin, ethurine, sulfactin, etc. Let's see the and last this is the disease, that is potato soft rot. This is caused by this pectobacterium. In that case, 52% of again this 52% of bacillus is reported against this as a PBB against this pectobacterium. And see that there is a certain type of reduction, 86% reduction, as well as
Sir,
100% reduction, and there is a
sir,
full
excuse
period of
me.
storage
We
because
have
this is a
1 minute left,
storage in
please.
disease. So Bacillus subtilis and Bacillus
Sir,
pumila,
please
this type of
continue
strain
within
is
1 minute.
full period of their their storing capability. And then we come to the mechanism of this soft lock. We see that when we use this plant beneficial bacteria, this bacteria produce the lipopeptides as well as volatile organic compounds and antimicrobial metabolites. Certain antimicrobial peptide synthesis gene also express upregulatively. This is the surfactant synthesis, A-fengicin, this type of enzyme. And as well as there is certain type of physiobiochemical and molecular changes in this plant, such as PAL expression, polyphenol oxidase expression, peroxidase expression. And in this pathogen, they downregulate the growth, motility, and biofilm formation, biodegradation of this N-acetyl homoserine lactose, and as well as there is a downregulation of polygalacturonase gene and cysteine gene. Okay. So this is the conclusion of my study is phytobeneficial biocontrolling bacteria represents sustainable biocontrol agent, and it is a reducing the dependence on chemical and bactericides in potato disease management. And as well as these are major bacterial diseases, it can be effectively suppressed by this diverse PBB. And there is a Bacillus, Pseudomonas, Streptomyces, the key biocontrol genera, and as well as antibacterial activity is mediated by the production of lipopeptides and secondary metabolites like surfactin, ethurine, fengicin, bacillicin, etc. An additional mechanism by the suppression of pathogen virulence gene by down-regulation of taxomin A biosynthesis gene in case of streptomyces, which is caused by the common scab disease. And the future perspective, another type of bacterial disease also can be checked as the biocontrol efficacy, and the molecular disease suppression mechanism. 3-season field trial is also needed for this future prospect, and the lyophilized multi-strain consortium is developed for the commercialization of this aspect. And this is the reference of my study, and this is thank you. And I also acknowledge my two collaborative universities, especially Panchavan University and Uttar Banga Krishi Vishwavidyalaya. Thank you.
Thank you, sir. Now, any question from the audience? No? Okay, again, thank you. Then the next presenter, our next presenter, Mrs. Monisha Mukherjee. Are you there? Monisha Mukherjee?
Yes, ma'am. I'm joining through my laptop. Can you give me a second? I'm joining through my laptop.
Yes, yes, please. You will be delivered
Okay,
on
thank you.
metabolic profiling
Thank
and enzymatic characterization on microbial isolates. Please proceed, madam. 20. 20. OP 28. Madam, are you ready for the presentation? Monisha, madam.
I'm joining my brother's account. Can you access? I have sent the meeting request. Ma'am, I have sent the request.
Let me check. Yes. Now you can proceed. Madam, are you ready? নাকি আপনি পরে দিতে চান? So for the time being, we'll move on to our next participant, uh, Mrs. Navanita Vishwas. Oh, sorry, sorry. It's okay, it's okay. Monisha madam will be presenting.
Ma'am, I have already shared my screen. Can you hear me? Am I audible?
Yes, yes, you are audible. Please proceed.
Is my screen visible?
Yes, now it is visible.
Okay.
Please close the small window.
Good afternoon, ma'am, and all present here today. I am
Ma'am,
Monisha
please,
Mukherjee, a
please
third year.
close the small window. Right hand side, a small window. Please close the small window. Or minimize it. Yes, now it's okay. Please go ahead.
Good afternoon, teachers, and all present here today. I am Monisha Mukherjee, a third year student of microbiology department. My topic is biochemical profiling of indigenous soil bacteria for heavy metal bioremediation. Okay, then bioremediation. Bioremediation is a biotechnology. In biotechnology, is a process where we use living organisms to detoxify contaminants. So, ma'am, can I proceed in Bengali?
Yes, you can.
সো বায়োরেমেডিয়েশন হচ্ছে এমন একটা বায়োটেকনোলজিক্যাল প্রসেস যেখানে আমরা লিভিং অর্গানিজমস গুলোকে ইউজ করছি, যেমন কি মাইক্রো অর্গানিজমস আমাদের কন্টামিনেন্টস গুলোকে ডিটক্সিফাই করতে। যেমন ফর এক্সাম্পল আমরা আমাদের প্রজেক্টে ইউজ করেছি এখানে কন্টামিনেন্টস হিসেবে মেটালস, lead, cadmium, arsenic, antimony। এই মেটালস গুলোকে আমরা ইউজ করেছি, আর সেখানে যে মাইক্রো অর্গানিজমস গুলো আমরা মানে আইসোলেট করেছি সেই সয়েল থেকে, আমরা দেখছি যে সেই মাইক্রো অর্গানিজমস গুলো কতটা কিভাবে মেটালস গুলোকে ইনকর্পোরেট করছে আর মেটালস গুলোকে লেস টক্সিক বানাচ্ছে। এন্ড এটাই আমাদের প্রজেক্টের মানে মেইন প্রজেক্ট, যেটা আমরা so পেয়েছি ফ্রম দি ইসকো স্টিল প্ল্যান্ট বার্নপুর। দেন সেখান থেকে সয়েল কালেক্ট করে সেটাকে আমরা ওয়ে করেছি, ওয়ে করে সেটাকে আমরা সিরিয়ালি ডাইলিউট করেছি ফ্রম 10 টু দি পাওয়ার -1 টু 10 টু দি পাওয়ার -7। দেন হায়ার ডাইলিউশন থেকে আমরা প্লেট করেছি, প্লেট করে পরে সেটাকে ইনকিউবেট করেছি ওভারনাইট। দেন কলোনিস গুলোকে সাবকালচার করেছি, দেন ওটাকে ফর পিওর কালচারস। দেন সেখান থেকে আমরা ওটাকে নিয়ে পরে 4 ব্যাকটেরিয়াল আইসোলেটস আমরা পেয়েছি, সেই 4 ব্যাকটেরিয়াল আইসোলেটস থেকে আমরা বায়োকেমিক্যাল অ্যাসে করেছি। দেন যদি আমরা বায়োকেমিক্যাল অ্যাসের কথা বলি, আমরা এখানে বায়োকেমিক্যাল অ্যাসে ফার্স্ট যেটা করেছি এটা হচ্ছে ইমভিক, দ্যাট ইজ ইন্ডোল, মিথাইল রেড, ভোক্স প্লাসকিউর এন্ড সিট্রেট ইউটিলাইজেশন টেস্ট। ইন্ডোল ইন্ডোল টেস্ট হচ্ছে এমন একটা টেস্ট যেটা আমরা করি দেখার জন্য যে ট্রিপ্টোফানিস এনজাইম ট্রিপ্টোফানটাকে কত ব্রেক করছে কিনা, ব্রেক করে সেখান থেকে ইন্ডোল ফর্মেশন হচ্ছে কিনা। ট্রিপ্টোফান ব্রথ আমরা এখানে ইউজ করি, যেখানে ট্রিপ্টোফান অ্যামাইনো অ্যাসিডে প্রেজেন্ট থাকে আর ট্রিপ্টোফানিস এনজাইম প্রেজেন্ট থাকে মাইক্রোঅর্গানিজমস এর মধ্যে, যারা কিনা ট্রিপ্টোফানটাকে ব্রেক করে ইনটু পাইরুভেট অ্যামোনিয়া অ্যান্ড ইন্ডোল। দেন ইন্ডোলটাকে আমরা যেটা পাচ্ছি সেটাতে আমরা কোভ্যাক্স রিএজেন্ট অ্যাড করছি। কোভ্যাক্স রিএজেন্টে হচ্ছে আইসোমাইল অ্যালকোহল, ডিএমএবি অ্যান্ড concentrated HCl. DMAB acid catalyzed reaction, যেখানে concentrated HCl সেটা অ্যাসিড মানে DMAB টাকে প্রোটনেট করে। সেই প্রোটনেট করার পর ইন দ্য প্রেজেন্স অফ দি অ্যাসিড সেটা ইন্ডোলের সাথে রিঅ্যাক্ট করে, যার জন্য আমরা একটা রোজ ইন্ডোল কুইনোডাল কম্পাউন্ড দেখতে পাই, যেটা হচ্ছে চেরি রেড ফর্মেশন রিং দেয়। যদি চেরি রেড ফর্মেশন রিংটা দেয় দ্যাট ইজ ইন্ডোল পজিটিভ, যদি না দেয় দ্যাট ইজ ইন্ডোল নেগেটিভ। যেমন কিনা আমরা এখানে দেখতে পাচ্ছি অবজারভেশনে, এখানে আমাদের একটা ব্রাউন ইয়েলো রিং ইন্ডিকেট করেছে দ্যাট ইজ ইন্ডোল নেগেটিভ। দেন আমরা যাবো তো মিথাইল রেড। মিথাইল রেড টেস্ট হচ্ছে আমরা এখানে করি আইটার ব্যাকটেরিয়া যেটা সেটা করে হচ্ছে মিক্সড অ্যাসিড ফার্মেন্টেশন। মিক্সড অ্যাসিড ফার্মেন্টেশন পাথওয়েটা যখন ওরা ফলো করে, দেন ওখানে এমআরভিপি ব্রথ এখানে আমরা ইউজ করি। সেই এমআরভিপি ব্রথের মধ্যে থাকে হচ্ছে গ্লুকোজটি করে ফার্মেন্ট করে এন্ড প্রডিউস করে হচ্ছে ল্যাকটিক অ্যাসিড, অ্যাসিটিক অ্যাসিড, ফর্মিক অ্যাসিড এন্ড সাকসেনিক অ্যাসিড। ফর্মিক অ্যাসিড ইজ ফার্দার ব্রোকেন ডাউন ইনটু কার্বন ডাইঅক্সাইড। দেন যেহেতু এতগুলো অ্যাসিড ফর্মেশন হচ্ছে সেই জন্য আমরা এটাকে মিক্সড অ্যাসিড ফার্মেন্টেশন বলছি। যেমন যদি আমরা এখানে দেখি, এখানে একটা রেড কালার ফর্মেশন এসেছে ডিউ টু দি প্রেজেন্স অফ দি ইন্ডিকেটর মিথাইল রেড। মিথাইল রেড যখন পিএইচ লো থাকে দ্যাট ইজ বিলো 4.4, তখন একটা রেড কালার দেখায় অ্যান্ড অ্যাবভ 6.8 ওটা একটা ইয়েলো কালার শো করে। ইট ডিপেন্ডস আপন দি কনসেন্ট্রেশন অফ দি হাইড্রোজেন আয়ন্স। দেন এখানে যদি আমরা দেখি ফর এক্সাম্পল যে পিকটা দেওয়া আছে এটাতে আমরা এটা ডিপিক্ট করতে পারছি যেটা হচ্ছে মিথাইল রেড পজিটিভ। যদি এটা মিথাইল রেড পজিটিভ হয় দ্যাট মিন্স আমাদের ভোজেস প্রস্টুয়ার নেগেটিভ হবে, কারণ আইটা আমাদের ব্যাকটেরিয়াটা অ্যাসিড ফার্মেন্টেশন পাথওয়ে ফলো করবে, বিউটিলিন পাথওয়েটা ফলো করবে। ভোজেস প্রস্টুয়ার এখানে বিউটিলিন পাথওয়ে যদি ফলো করতো দেন এটা ভোজ প্রস্টুয়ার পজিটিভ হতো। বিউটিলিন পাথওয়ে ফলো করলে ওটা হচ্ছে অ্যাসিডোইন ইন্টারমিডিয়েট হয়, সেই অ্যাসিটোন ইন্টারমিডিয়েটের সাথে আমরা ব্যারিজ রিএজেন্ট এ এন্ড ব্যারিজ রিএজেন্ট বি অ্যাড করি। ব্যারিজ রিএজেন্ট এ হচ্ছে 5% আলফা ন্যাপথল এন্ড ব্যারিজ রিএজেন্ট বি হচ্ছে পটাশিয়াম KOH এন্ড দেন ওটার সাথে ডায়াসিটাইল ফর্মেশন হয়। ডায়াসিটাইল ফর্মেশন থেকে আমরা কালার দেখতে পাই, যদি পিঙ্ক রেড কালার এসে থাকে দ্যাট মিন্স ইটস ভোজেস প্রসপের পজিটিভ, আর যদি না এসে থাকে দ্যাট মিন্স ইটস ইয়েলো ব্রাউন কালার, দ্যাট মিন্স ইটস ভোজেস প্রসপের নেগেটিভ। এখানে এই পিকচার আমাদেরকে ডিপিক্ট করছে ভোজেস প্রসপের নেগেটিভ। দেন সিট্রেট ইউটিলাইজেশন টেস্ট হচ্ছে এমন একটা টেস্ট যেটা আমাদেরকে এটা বুঝতে হেল্প করে যে সিট্রেট যদি আমরা এজ এ সোল কার্বন সোর্স হিসেবে ইউজ করি উইথ দ্য হেল্প অফ দি এনজাইম সিট্রেট পারমিয়েজ, দেন একমাত্র সিট্রেটটা পজিটিভ হবে। এখানে একটা ইন্ডিকেটর থাকে যেটা হচ্ছে ব্রোমোথাইমাল ব্লু, যেটা গ্রিন ইন কালার থাকে। তো সেটা একটু ব্লুইশ গ্রিন যদি হয়ে যায় দ্যাট মিনস ইটস সিট্রেট পজিটিভ, আর যদি না হয়ে থাকে দ্যাট মিনস ইটস সিট্রেট নেগেটিভ। And from this picture we can depict its citrate positive. So the symbol is negative positive, which indicates that it's enterobacter erosion. Then catalyst test. Catalyst test হচ্ছে একটা বায়োকেমিক্যাল assay যেটা আমাদেরকে এটা ডিটেক্ট করতে হেল্প করে যে হাইড্রোজেন পারঅক্সাইড প্রেজেন্ট আছে কিনা। যেমন ডিউরিং এরোবিক রেসপিরেশন অনেক টক্সিক বাইপ্রোডাক্টস প্রডিউস হয়, যেমন কি সুপার অক্সাইড র্যাডিক্যালস অর হাইড্রোজেন পারঅক্সাইড। আর সেখানে যখন আমরা ক্যাটালিস্ট টেস্ট করি, কিছু মাইক্রোঅর্গানিজমস থাকে যারা এই টক্সিনস গুলোকে নিউট্রালাইজ করার জন্য ক্যাটালিস্ট এনজাইম প্রডিউস করে। এই ক্যাটালিস্ট এনজাইমটা হাইড্রোজেন পারঅক্সাইডটাকে ব্রেক ডাউন করে দেয় ইনটু হাইড্রোজেন এন্ড অক্সিজেন। আর অক্সিজেন ফর্মেশনটাকে আমরা দেখতে পাই উইথ দ্য হেল্প অফ দি রাপিড এফারভেসেন্স, দ্যাট ইজ গ্যাস বাবল ফর্মেশন। যদি যখন পিকচারটা আমাদেরকে ডিপিক্ট করছে গ্যাস বাবল ফর্মেশন means it's catalyst positive, and now catalyst negative. Then protease activity. Protease activity depend এটা
You
depend
have
করছে
1 minute
যে
left only. Try to conclude within 1 minute, please. Please proceed. You have 1 minute also.
ma'am, I can't hear. Hello.
Hello? Am I audible?
You are not audible, ma'am. Please repeat
You can conclude
just you
your
said.
you can conclude your presentation within 1 minute, because our time limit is 8 minutes for deliberation and 2 minutes for question answer discussion. Okay? Already 7 minutes passed. So you have left only 1 minute. Try to conclude within 1 minute.
Okay. So ma'am, protease activity যেটা হয় যে মাইক্রোঅর্গানিজমস এক্সেসরিয়ালি এনজাইমস প্রডিউস করে, দ্যাট ইজ প্রোটিজ, যেটা কিনা প্রোটিনস থেকে ব্রেকডাউন করতে পারে ইনটু পেপটাইডস এন্ড অ্যামাইনো অ্যাসিড, যেটা থেকে ওটা সেলের মধ্যে ইনটু দ্য সেল মেমব্রেন ইনকর্পোরেট করতে পারে। যদি যেমন কি ইন দ্য পিকচার ইট ডিপিক্টস দ্যাট যদি এটা লিকুইড ফর্মে থাকে যেমন এটা আছে, দ্যাট মিন্স ইটস প্রোটিজ পজিটিভ, আর যদি না থাকে তো তাহলে এটা হচ্ছে নেগেটিভ। দেন অ্যামাইলেজ অ্যাক্টিভিটি। Amylase, we know it's a polysaccharide which is made up of amylose and amylopectin. Amylase the structure it is bonded by alpha 1,4 glycosidic bond. Amylase একটা enzyme যেটা produce করে কিছু microorganisms for incorporating it into the cell membrane. অ্যামাইলেজ যখন প্রডিউস করছে তো ওদের মধ্যে সেই বন্ডটা ওরা ব্রেক করছে। এবার স্টার্চের একটা হেলিক্যাল স্ট্রাকচার থাকে, এটাকে আমরা ভিজুয়ালাইজ করেছি উইথ দ্য হেল্প অফ দি লুগলস আয়োডিন। যখন লুগলস আয়োডিন আমরা দিই দ্যাট মিন্স তখন ওখানে যেহেতু স্টার্চের একটা হেলিক্যাল স্ট্রাকচার থাকে সেখানে আয়োডিনের যে আয়ন্স গুলো সেগুলো ট্র্যাপ হয়ে যায় বলে একটা ব্লু ব্ল্যাক কমপ্লেক্স হয়। যেমন ইন দিস পিকচার আমরা দেখতে পাচ্ছি একটা গোল্ডেন ইয়েলো ফর্মেশন হয়েছে বাই স্ট্রিকিং, যেটা আমরা এটা ডিপিক্ট করছি যে এখানে অ্যামাইলেজ অলরেডি ব্রেকডাউন হয়ে গেছে, অ্যামাইলেজ ব্রেকডাউন করে দিয়েছে বলে এখানে গোল্ডেন ইয়েলো ফর্মেশন হয়েছে, দ্যাট মিন্স ইটস অ্যামাইলেজ পজিটিভ। বাকি যে এরিয়াটা ইট ইজ কভার্ড বাই ব্লু ব্ল্যাক কমপ্লেক্স দ্যাট ইজ লুগলস আয়োডিন। And result আমরা দেখতে পারি যে indole test it's negative, methyl red positive, Otis plus Q negative, secretization positive, catalase test negative, protease activity negative, and amylase test positive. And then এটাই আমরা conclude করতে পারি যে যদি biochemical assay, its environmental sustainability, in situ treatment, cost effectiveness, addressing heavy metal contamination যেটা আমাদের main for the bioremediation project, versatility across pollutants. Thank you.
Thank you, ma'am. Any question from audience? No question. So thank you again.
Thank you, ma'am.
Is there any question?
Test which heavy metal you have used in your
Ma'am, one question appears.
Okay. We have used four heavy metals: lead, cadmium, arsenic, and antimony.
Okay, thank you.
Okay, thank you.
Thank you, sir.
Now we will move on to our next presenter, Ms. Nobonita Vishwas. Are you there? And Godfrey Dotto. Navonita Vishwas or Gokuli Dotto.
Yes, ma'am, I'm here.
Okay, okay. You will be delivered on the topic characterization of microbial resistance via MIC and agartha diffusion. So
Yes, ma'am.
please proceed. OP 30.
Ma'am, I, ma'am, I'm sharing my screen.
Okay. Please proceed. You will have 8 minutes for your
Is
presentation.
my ma'am, is my screen visible?
Yes, it is visible. Please proceed. You have 8 minutes for your deliberation. Okay? Please make it full screen.
Hello.
Okay, okay. Now it is okay. Please proceed.
Thank you. Good evening, everyone present here. I am Navnita Vishwas, a third year student of microbiology. Today my topic is characterization of innovation in system at MPC. Okay. হচ্ছে
Now it's okay.
ছিল আমরা
Have you opened any other devices?
প্রেজেন্ট থাকি
Please use one devices
Yes,
only.
ma'am.
Otherwise, your voice is cracking. It is not audible properly. Please join from one devices
It's not
only.
working.
Please mute another devices. Log in into one device only. Otherwise, your voice is cracking, not audible properly. Navonita or Gokuli মোবাইল বা অন্য কিছু অন থাকলে সেটা অফ করে যে কোনো একটা ডিভাইস থেকে জয়েন করো প্লিজ জয়েন টু ওয়ান ডিভাইস অনলি otherwise your voice is cracking
Guys, আমি একটা ম্যাম
your your word is no your voice is not audible properly the sound is cracking Navanita, are you there? Navanita or Rupali, are you there? Navanita or Rupali, are you there? Otherwise, we have to proceed for another presentation. Navonita or Gozuli, are you there? We have another presentation, so if you ready, then possible.
ম্যাম আমার ম্যাম আমার নেট কানেকশন অনেক এরর দিচ্ছে একটু
কি হয়েছে? কি সমস্যা? কি সমস্যা? এখন কি দেওয়া যাবে প্রেজেন্টেশন নাকি আমরা নেক্সট পার্টিসিপেন্টকে কল করতে পারি? Navonita, are you ready? So we will move on to our next presentation. So now the next segment of this online presentation is conducted by.
হ্যালো ম্যাম ম্যাম আমার ইন্টারনেটটা আমার প্রচুর প্রবলেম করছে একটু 2 মিনিট দেওয়া যাবে ল্যাপটপে কানেক্ট হচ্ছে না
তাহলে তুমি দেখো একটু পরে যদি চেষ্টা করে পরে
আচ্ছা ঠিক আছে
চেষ্টা করো।
থ্যাংক ইউ ম্যাম
Now the next
ঠিক আছে ম্যাম
segment of
থ্যাংক
this
ইউ
online presentation will be conducted by Dr. Pradeep Bhati sir. I request Dr. Pradeep Bhati to take over the charge. Please.
Welcome to the session. In this session, there are 4 papers to be presented. First one is topic 33, presented by Sudarshan Nandi. I think you are there.
Hello. Hello. Am I audible, sir?
33. Yeah. Your presented topic is comparing different fish fundamentals and techniques in northeastern part of India. So, Mr. Sudarshan, please start your presentation. 10 minutes already
Yes,
for
sir.
presentation, 3 for presentation and 2 minutes for question and answer. Please start your presentation.
Okay, sir. Thank you so much. My slides are visible. Hello.
Yes.
Can you hear me? Okay. Thank you so much. So today, good afternoon all. Today I'm going to talk on comparing different fish fermentation techniques in northeastern part of India. So what is fermentation? First, I would like to address the fermentation is a metabolic process that obtains energy from organic compound without using any external oxidizing agent. And it is not a very new technique, it is an ancient one where it is actually great for preservation method and where the microorganisms are actively involved. This technology, which is safe for environmental friendly as well, and it requires poor energy consumption and present, and it is also actually very much useful for the future use. So this is the evidence that it is an ancient practice, and the fermentation can be done to preserve many types of food products as well. So now coming to the fermentation in the mode of fish fermentation. So fish fermentation is a popular traditional method which followed in the northeastern part of India, which is my targeted site And here actually that work has been done, the northeastern part of India. Here the fish fermentation is a popular method. So the people of this part, like Nepal, Bhutan, Darjeeling, then Sikkim, Assam, Arunachal Pradesh, and many more, these states are actually often they are doing the fish fermentation process. So the motto or the main aim of this fish fermentation is actually uh, means because of the fish, all the fish species are not available throughout the year. So this is one very important method to preserve food, and whenever they want, they can consume it. Additionally, there is also added benefit which can enhance flavor, increase digestibility, as well as it is actually improving therapeutic values as well as nutrient upliftment. With this, in the northeastern part of India, the fish fermentation is carried out through means generation after generation. Means it is a practice in household level, and it is done means community level as well. So with this, I just like to show the means science. So this is the fish means whenever we are talking about fish fermentation, definitely there will be definitely there will be production or added benefit like free amino acid will be increased, then biogenetic amines will be increased, oxidant that means minerals, PUFAs, these things will be means increased in the food product, and where the active involvements are there are governed by the microorganisms. So in the northeastern part, why I have taken this as my study field as because it is a hotspot where we can see various type of small indigenous fish species are there. So these are the very common species where we can see there are very diversified species, and here, these fish are actually often go for the fish fermentation process, like the Polyphlongodon, Mola, Puntius, Chana, then Mistras, Vitas, all these small fish are actually occasionally or eventually they are going for the fish fermentation. So before proceeding, let me just focus to the main objective of the study to compare nutrient enrichment of SIFs with the large carp. And the second objective is to compare the different fish fermentation process of the SIF of naive NEF means the northeastern part of India, and to check size-wise nutrient enrichment of Amyliplanodon mola, which is a very important or popular small indigenous fish species. With this, the first objective is the result of the first objective, where we can see that SIFs are having more nutrient enriched one. If we are comparing with the big fish like the carp, katla, hilsha, so the small fish categories like Amyliplanodon, Isopus, Ranticus, they are having more vitamin A, calcium, and iron content in their body. So now let me go for the second objective. So the first, I have explored the first one, which is the Shidal, which is done, one of the very important fish fermentation process. So it is done in the targeted fish like Puntius, Amyliplanodon, and Mola, and it is actually very much used in the especially states of Tripura, Assam, Mizoram, Arunachal Pradesh, especially in the state Tripura and Assam. So if we look after the process, how they are doing, traditionally how they are doing, so this is the process where the shidal, means how the shidal actually means used to prepare. So the fish species like Amyliplanodon and Mola used to go for the sun drying process after certain days, like 2 to 5 days for the Amyliplanodon and Mola. So it will be going for processed, more further processed. Here, very interesting part is actually done through the process of the production of the ardent pot. So here we can see ardent pot is actually produced before going to the further. So oil actually here the ardent pot is made up will applied with the mustard oil, and it will be done through until it reach its saturation point. So it will be very important process, steps of the fish fermentation. Then afterwards, this sun-dried fish will be again going for water cleaning, and afterwards it will be done through shopping, and then packing will be done. So the fourth step is also as well is very important. So in this earthen pot, the packing will be done, and the tight packing will be done, and lastly sealing of the pot. So here in the interior portion of the earthen pot, sealing is done with the use of the dried fish. There will be packed with the dried fish, which will be actually help for the microorganism grow. So here the microorganism will be growing and spreading with all over the pot. So here afterwards, this pot will be kept under the mud, under the soil, you can tell, or ground. So this, and 1/3 will be in the outside of the ground. So then it will be after 3 to 5 months, the will be prepared. So this is the details of the microorganism. After normally at least 120 days, the seedle will be produced, and these are the microorganisms which will be involved. Coming to the next one is the nari, which is actually very popular
Madam,
for
please
fish
conclude
farming.
your talk in 1 minute.
Sorry, sir.
Conclude your talk in 1 minute. Because time over.
I am audible or not, sir?
You have 1 minute.
You have 1 minute left. Please conclude within 1
Okay,
minute.
okay. So this is also a very important process of the fish fermentation, which is done in Manipur. So similar kind of fish fermentation process is used for this nari. But here, one interesting part, that earthen pot will be not kept inside the ground. It will be kept means in normal, in ambient temperature. And after 4 to 12 months, the fermented fish product will be produced. So, and these are the details of the microorganism. Now coming to the next one, that is a tungtap. So here it is actually done in Meghalaya, particularly Khasi tribe used to do it. So here, interestingly, same thing is actually followed, but here additionally, they are using salt. So the fish will be mixed with salt, and further fermentation process will be done, and it requires 6 to 7 months. And again, it will be done in the earthen pot, and afterwards we can see these are the microorganisms which are involved in this process. So lastly, the hentak, which is also a very important fish fermentation and process which is done in Manipur, and here we can see the fish is almost dendricus will be mixed with the plant, the alocasia plants with these two will be mixed together, and the fish fermentation product will be actually made. So you can see the plant mixture, and this is the fish mixture. So those two will be kept like 1:1 ratio, and after 2 weeks it will be actually prepared. So these are the details of the microorganism. Now coming to the last point, or the last objective. So this study reveals that if we check the Amblyopharynga and mola, which is a very important small indigenous fish species, their nutrient enrichment varies according to the size. So if we are talking about the small size group, so it is more nutrient enriched. If we are going for 8 to 16 centimeter, so it is less, means comparatively less. So we'll suggest to focus the small size SIF to get more beneficial nutrients, and then we can go for the fish fermentation with the small size pigs. So this is all. So for my conclusion, fermented fish products can be used for long time, and which are actually very important for their adding more nutrients to the society, and which can be combat with the malnutrition like problem. Various fish fermentation techniques are there in the northeastern part of India. Many are relatively unexplored, so there are many work to be done. And this is actually the technique which is low cost, added economic is empowered to the tribal and the rural society. With this, I would like to conclude. Thank you all.
Okay, thanks for your nice presentation. Now, any questions from audience? Audience from online, you can also ask any questions. Okay, thanks again. I think there is no questions from audience. So we go to the next presentation. Is OP40 by Dipalita Sinha. I think, madam, you are there. Yeah.
Yes, sir, I'm there.
Okay, so madam, you are presenting characterization of Candida tropicals strain with altered into microbiomes. So please start, madam, your presentation. You are allotted 8 minutes for your presentation and 2 minutes for question and answer.
Sir, is my slide visible?
Yes.
Okay, sir. Good afternoon, everyone. I am Dipanita Sinha, and my work is titled Characterization of Candida tropicalis strains with Altered Endomicrobiomes. Yeasts are unicellular fungi found all over the earth, including in the atmosphere, soil, water, and even in human bodies. They are known to perform various roles, such as decompositions, performing mutualistic or competitive interactions, and may even serve as pathogenic to other organisms. They are also hosts to varieties of microbes known as the endomicrobiota. This endomicrobiota may affect the yeasts in either positive way or negative way, as given in the example. Among these yeasts, Candida tropicalis is known to harbor a variety of intracellular bacteria such as Staphylococcus, Chrysobacterium, etc. Even Helicobacter cells were discovered in this Candida tropicalis. The JY101 strain of Candida tropicalis was reported to contain Deinococcus thymus phylum and unclassified Pseudomonas reeds, which were unique to this particular strain of Candida tropicalis only. My study focuses on the comparative analysis of Candida tropicalis JY101 strain, which was treated with antibiotic fosmidomycin JY101B and was further treated with antibiotics gentamicin and rifampicin JY101 delta B against the wild strain. Major experimental variables assessed were growth kinetics, different substrates supporting growth, and the toxicology of the strains in the presence of bacterial as well as fungal antibiotics. Moving on to the experiments, first we come to the physiological characterizations. Initially, the three strains of Candida tropicalis were revived from the glycerol stock and then cultured overnight in fresh YPD media. One of the flasks was marked as blank, that is, it was uninoculated, and two flasks each were inoculated for the three respective strains. OD readings were taken at 620 nanometer at 2-hour intervals. The final reading recorded was the average of the duplicates for each strain. Going to the biochemical characterization, a carbon substrate utilization assay was done by taking a basal media which lacked carbon source but contained all other nutrients. The carbon source substrates were chosen as glucose, a certain fatty acids, and certain aromatic hydrocarbons. In this experiment, 7 boiling tubes were taken. One was uninoculated, 3 for the strains grown in carbon source, and 3 for the strains grown without carbon source. They were incubated at 30 degrees Celsius at 140 to 150 RPM, and OD readings were taken at 620 nanometers at the 0, 20th, and 40th hour marks to calculate the growth yield. In the toxicological characterization, there were two parts, one using the fungal antibiotic cycloheximide and the other using the disc diffusion assay by using various bacterial antibiotic discs of different concentrations. These preliminary tests showed anomaly recording the tetracycline inhibition, which was further verified by the disc diffusion and also a growth curve based confirmatory test. Coming to the results, we see that under standard growth conditions, the three strains exhibited a typical sigmoidal curve, showing certain differences. In the substrate utilization assay, by the students of Canada Tropicalis, we see that JY101 delta B has the maximum growth in case of almost all substrates except for benzoic acid. And the bacterial antibiotic assay, see that JY101B shows a zone of inhibition around the tetracycline disc after 24 hours, which disappears after the 48 hours. By performing the confirmatory test, we see that not only JY101B but also delta V shows a zone of inhibition after 24 hours, which disappears after 48 hours. In the growth curve test, we see that there is a prolonged lag phase in case of both JY101B and delta B, but B achieves the comparable steady state stationary OD value, while delta B shows a marked decrease in the stationary state OD value. The fungal antibiotic cycloheximide could not inhibit either of the three stains. Coming to the conclusion, we see that since JY101 delta W had immediate growth and highest stationary state OD value, it would mean that the stable microbiome supports the growth of the strain. But B reached the stationary state earliest, so since it has been treated with antibiotic phosphorylomycin, we can say that the competitive microbiome has been disrupted. And delta V shows a prolonged lag phase due to extensive microbiome alteration by triple antibiotic effect, but it has ultimately achieved the stationary phase comparable to the wild type, indicating potential physiological compensation. The carbon neutralization assay shows that the JY101 delta B has highest growth and fatty acids, likely due to effective removal of competing endomicrobiota, whereas JY101B shows the lowest growth due to impaired but persistent endomicrobiota, as we know that phosphomycin blocks the MEB pathway in certain bacteria, thus leaving them impaired. And JY101W shows a baseline utilization in case of all. In the toxicological evaluation, a transient tetracycline sensitivity in both JY101B and delta B proves that the composition of the entomicrobiota is a fundamental aspect in yeast inhibition by bacterial antibiotics. In JY101B, Brucella species were found, and we know that phosphomycin inhibits growth of Brucella species. But most likely, they recover from this inhibition after a certain period of time. So, JY101B doesn't show any decrease in the stationary state OD value, while delta B does not fully regain its normal growth and hence has a marked decrease in its stationary state OD value. For fungal antibiotic, there was no inhibition. From these results, we can infer that the endomicrobiome of the yeast definitely affects its functioning. Future work targeting these endomicrobiota by manipulating them could help in metabolizing toxins in the human body. Also, identification of the exact endosymbionts responsible for the antibiotic resistance, and by removing them, we can make the yeast susceptible to the drug or the antibiotic. So, these are the references. Acknowledgement. Thank you.
Thank you, madam, for your in-depth presentation. I expect few questions from audience. From online participants also. Any question? No. Okay. Thank you, madam, for your presentation. And now for the next presentation, OP2 by Sunandana Mandal. I think madam is there. Okay.
Yes, sir. Am I audible?
Yeah, you are audible. And you start your presentation. You will deal the
Yes,
effect of
sir. Actually, from today morning, I'm having some problem in my laptop. So, can I present through my phone?
Sure, no problem. Please.
Okay, thank you so much. Um, am I audible, sir?
Yeah.
Hello?
Yes, you're audible. Please try to maximize your skill.
Hello?
Yeah, you're audible.
Hello, sir, am I audible?
Yeah, you're audible. Please start. Any issue?
Hello, am I audible, sir?
Yeah, you're audible. Please start your presentation.
Hello?
Yeah, you're audible.
Hello? Hello, sir, am I audible?
Yeah, you're audible. Please start.
Am I audible, sir?
Yeah, you're audible. Please start.
Hello, am I audible, sir?
Yes, you're audible. Start your
So,
presentation, please.
yeah, sorry, sir. I'm just delivering my lecture, okay? Because there is some problem with the slide presentation.
Okay, please.
Okay. Yeah, okay. Respected chairperson, distinguished faculty members, my fellow participants, very good afternoon to everyone. I am Dr. Shunandana Mondal, currently working as an assistant professor in the Department of Chemistry, Moina College. The topic of my today's presentation is elucidating the impact of zinc fertilization on soil dynamics within rice cultivation in red acidic soils of eastern India. So, zinc deficiency has been identified as one of the major concerns in the current scenario, resulting in severe health complications. And this has been found to be severe for the cereal eaters, especially rice and wheat. Improvement of the nutritional quality of rice grain thus appears as a solution to this problem. Zinc as an essential micronutrient for the growth, yield, and quality of the rice crop. Application of zinc fertilizer thus can be considered as an attention to correct zinc deficiency in rice grain. So the goal of my study is to evaluate the effects of zinc fertilization on different zinc fractions present in soil and to determine their response to rice crop. So for this we have carried out our experiment during two times, that is Kharif and Rabi season, and we have carried out the sequential fractionation procedure for determining different zinc fractions present in soil. So first we see that the DTPA extractable zinc concentration in soil varied significantly with zinc fertilization over the control, and the highest DTPA zinc were recorded in T5, and higher zinc content in soil with zinc fertilization was recorded in treatments receiving zinc sulfate heptahydrate in two equal splits in comparison to its single vessel application. So with advancement of time, the availability of applied zinc in soil decreases. So now we come to the water soluble and exchangeable zinc, and we see that its value also increases with zinc application, and this contributes least to the total zinc value. And water soluble and exchangeable zinc has high mobility and lability, hence readily available to plants. The next fraction is the organically complexed zinc. This is also a very this contributes least to the total zinc fraction. Then we come to the crystalline or amorphous sesquioxide bound zinc fractions, which is related directly to the iron oxide content of the soil. So zinc present in comparatively labile pool, what happens? It gets absorbed on the surface of these oxides. The value of zinc present in this fraction increases over time. Higher amounts of crystalline sesquioxide zinc present in soil solution suggests that the crystalline iron oxides have greater ability to absorb zinc than that of the amorphous sesquioxide zinc. Soil zinc application significantly increases this fraction also over control. The next fraction is manganese oxide bound zinc, and manganese or iron oxides are considered to be the heavier reservoir of heavy metal. So concentration of zinc in different fraction decreases over time in the plots treated with no zinc fertilizer, which can be due to the removal of different zinc fractions by crops. So the next one is the total fraction, total zinc. So this is total zinc actually is governed by factors like cropping system, minerals present, and climate conditions. So it is the parent material that determines the content of total zinc in soil. So we have already analyzed several parameters present, there's several zinc fractions. So soil parameters are considered to be one of the main governing factors to determine the phytoavailability of an element. So an inverse relationship exists between soil pH and water soluble and exchangeable zinc, which means that at higher pH more hydroxyl ions are present in soil solution, which causes the precipitation of zinc hydroxide. Water soluble zinc and organically complex zinc are considered to be the two important fractions that are major contributors towards available zinc. DTPA zinc is slowly released from different zinc fractions, which is evidenced by the existence of significant and positive correlations between them. And with time, we see that easily the extractable fractions get transformed into stable fractions. The significant and positive correlation between soil organic carbon and water soluble and exchangeable zinc suggests that exchange sites are provided by the soil organic carbon for adsorption of zinc, which prevents the fixation of zinc by soil components. So then we have the correlation heat map, which is the visualization of the different color patterns of the color coded matrix revealed a strong positive correlation between different zinc fractions, which actually indicates that zinc present in one fraction can get easily converted to the other fractions over time. Zinc fertilization on foliage results an increase in grain zinc content due to enhanced protein biosynthesis, and it appears as an effective solution to avoid the complex reactions of soil applied zinc with various soil components. Enhancement in grain zinc content was recorded when zinc sulfate was applied in split in comparison with single vessel application over time. Zinc gets precipitated as zinc carbonate or zinc sulfide, which decreases its availability. Thus, split application always performs better in comparison to the single vessel application. So next we have discussed the multiple regression equation, and finally we can conclude that greater contributions are coming from the labile zinc pools. Thank you so much for your patience, and I once again apologize for from my end. Sorry, very sorry. Sorry, sir, I cannot hear.
Okay, too. Thanks for your nice deliberation. And now, any questions
Thank
from
you, sir.
audience?
Ma'am, I fixed my problem. Am I audible?
Yeah, you're audible.
May I proceed, ma'am? I am Navneeta Biswas. Can I proceed?
One presentation I left in the previous session is Nobody Vistas.
Yeah, yes, sir.
You're there?
Yes, sir. I'm there.
Okay, you start your presentation.
Okay, sir. Is my screen visible, ma'am?
Yeah, please go on. Please.
Ma'am, am I audible?
Yeah, audible. Please continue.
Uh, Good evening, everyone. Everyone present here. I am Navneeta Vishwas. I am a student of Asansol Gaskora. I am a third year student. Today, my topic is characterization of microbial resistance and growth inhibition in the bioremediation system via my MIC and agar curve description. Bioremediation is a sustainable and eco-friendly approach that utilizes microorganisms to degrade environmental conditions. That effectiveness of the process largely depends on the metabolic resilience and tolerance of microbial strains towards pollutants and co-contamination such as heavy metals. To evaluate to evaluate microbial resistance and growth inhibitions, standardized techniques like minimal inhibitory concentration and agar-cut diffusion assay are employed. These methods help determine the tolerance themselves and assay impact on the contamination concentration on microbial growth. Understanding these sustainability patterns is essentially for selected level microbial isolation capable of surviving toxin environmental via maintaining effective degradation effective. Okay. Can I continue in Bengali, please? Sir?
Yes.
Sir, am I audible?
Yeah, you're audible. Okay, you're audible. Please continue.
Ma'am, am I audible?
Yeah, you're audible. Please continue.
ম্যাম আমি বাকিটা বাংলায় কন্টিনিউ করছি।
Okay, fine. Bangladesh.
তো বেসিক্যালি আমরা একটা জায়গা থেকে সয়েল স্যাম্পল কালেকশন করেছিলাম। সয়েল স্যাম্পল কালেকশন করার পর ওটাকে আমরা সিরিয়াল ডাইলুশন করেছিলাম। সিরিয়াল ডাইলুশন করার পর ওখান থেকে আমরা ওটাকে পর প্লেট এবং স্প্রেড প্লেটের মাধ্যমে 4টি ডিফারেন্ট টাইপ অফ ব্যাকটেরিয়া ওখান থেকে কালেক্ট করেছিলাম। বেসিক্যালি আমরা বায়োরেমিডিয়েশনের প্রসেসটাকে কন্টিনিউ করার জন্য আমরা ডিফারেন্ট টাইপ অফ হেভি মেটালস ইউজ করেছি এখানে, যেমন হচ্ছে লেড, ক্যাডমিয়াম, আর্সেনিক আর অ্যান্টিমানি। ফার্স্ট স্টেপ আমরা করেছিলাম আগার কাপ। আগার কাপে আমরা বেসিক্যালি নিউট্রিয়েন্ট ব্লড ডাইলুশন করেছিলাম। নিউট্রিয়েন্ট ব্লড নেওয়ার পর আমরা প্লেটিং করেছিলাম। প্লেটিং করার পর আমরা যে সয়েল স্যাম্পল থেকে যে ব্যাকটেরিয়াগুলো আমরা পেয়েছিলাম, সেই ব্যাকটেরিয়াগুলো আইসোলেট করার পর সেগুলোকে অ্যাজ আ ইনাকুলাম হিসাবে আমরা আগার কাপে দিয়েছিলাম। আগার কাপে দেওয়ার পর আমরা ওদের ডিস্ক কাটার পর সেখানে আমরা মেটাল সাসপেনশন অ্যাড করেছিলাম। 4টে ডিফারেন্ট টাইপ অফ মেটাল সাসপেনশন আমরা বানিয়েছিলাম ইউজিং ওই মেটালগুলোকে হেভি মেটাল নিয়ে ইউজ করার পর আমরা 4টে ডিফারেন্ট টাইপ অফ মেটাল সাসপেনশন ওগুলোতে অ্যাড করেছিলাম। অ্যাড করার পর আমরা 24 আওয়ার ওটাকে ইনকিউবেশনে রেখেছিলাম। অ্যাজ আ রেজাল্ট হিসাবে আমরা দেখেছি যে আমরা যে আগার টিপগুলো বানিয়েছিলাম সেগুলোতে কিছু কিছু জায়গায় জোন অফ ইনহিবিশন দেখা গেছে, মানে কিছু কিছু ব্যাকটেরিয়া যেগুলো আমরা পেয়েছিলাম ওগুলো থেকে, সেগুলো কিছু কিছু যে ব্যাকটেরিয়াগুলো আমরা সেখান থেকে পেয়েছিলাম যেগুলো কিছু কিছু জোন অফ ইনহিবিশন দিয়েছে, কিছু কিছু ওভারল্যাপ করেছে এবং বাকি কিছুটা লেস টক্সিন হিসেবে আমরা দেখতে পেয়েছি এখানে। এটা অ্যাকচুয়ালি ছিল প্রাইমারি স্টেপ অফ বায়োরেমিডিয়েশন। নেক্সট আমরা করেছিলাম হচ্ছে কি যে এমআইসি, যেটা হচ্ছে ব্লড ডাইলেশন মেথড। Basically, it had a lowest concentration and antimicrobial agent যেটা prevents করে visible growth of microorganism after overnight incubation. ফার্স্টে আমরা এমআইসি স্টেপ হিসেবে আমরা করেছিলাম একটা নিউট্রিয়েন্ট ব্লথ প্রিপেয়ার করেছিলাম। নিউট্রিয়েন্ট ব্লথ প্রিপেয়ার করার পর আমরা ওটা 12টা টেস্ট টিউব নিয়েছিলাম। 12টা টেস্ট টিউব নেওয়ার পর সেই নিউট্রিয়েন্ট ব্লথগুলোকে আমরা 4টে ডিভাইড করেছিলাম। ডিভাইড করার পর তারপরে সেম অ্যামাউন্ট অফ আমরা হেভি মেটাল ইউজ করেছিলাম অ্যাজ আ অ্যান্টিবায়োটিক আর অ্যান্টিমাইক্রোবিয়াল এজেন্ট হিসাবে, আর ইনাকুলাম অ্যাড করেছিলাম। ইনাকুলাম অ্যাড করার পর আমরা ইনকিউবেশন করেছিলাম এবং ডাইলিউশন করেছিলাম। ডাইলিউশন করার পর আমরা ওটাকে ওভারনাইট ইনকিউবেট করেছিলাম। ইনকিউবেশনের পর অ্যাজ আ রেজাল্ট হিসেবে আমরা পেয়েছি যেগুলো বায়োফিল ফর্মেশন হয়েছে, যেগুলো সেগুলো অ্যান্টি টক্সিসিটি শো করেনি, আর কিছু কিছু ক্ষেত্রে সেগুলো শো করেছে। এখানে আমি ওডি ব্যাখ্যা করতে পারি। রেজাল্ট আমরা দেখলাম যে বায়োরেমিডিয়েশন দোকানে প্রসেস করেছিলাম, সেখান থেকে আমরা রেজাল্ট পেয়েছি। এর মধ্যে দেখতে পাচ্ছি যে আর্সেনিক, অ্যান্টিমনি আর এর সব থেকে টক্সিক হচ্ছে, মধ্যে সব থেকে বেশি টক্সিক যেটা শো করেছে সেটা হচ্ছে অ্যান্টিমনি। টক্সিসিটি সব থেকে বেশি এবং সব থেকে লেটেস্ট হচ্ছে ক্যাডমিয়াম। সব থেকে লিস্ট যেটা শো করেছে সেটা ছিল ক্যাডমিয়াম। And last in the competition প্ল্যাটফর্ম করেছিলাম, সেখানে আমি কম্পিটিটিভ ডিফারেন্ট টাইপ অফ ইউজ করতাম, কতটা বেশি কতটা সেটা আমরা জানতে পেরেছি। থ্যাংক ইউ।
Thank you,
থ্যাংক ইউ ম্যাম।
Okay, thank you, Listen. Now this is the time for question and answer. If any question from audience, I think there is no questions, so thanks again. So now I conclude this session and hand over to the.
Thank you. আর গোটা স্লাইডে কি দিতে হয়, এটা পুরোনোটা।
Thank you,
ma'am.
Thank you all the participants for your excellent presentations. We are truly delighted here in Asansol Girls College. All your path-breaking research is truly going to augment the research in biosciences. It is truly, we are humbled and thrilled and excited that many young, UGM PG students, they have taken up a very innovative experimental strategy to showcase their research here in this international conference. And we are truly honored that all the young minds, they thought that they should come to Asansol all the way from Kolkata and different parts of the country and it's a great privilege to hear your new ideas and the way you have presented here in Asansol Girls College. We are truly honored, and this is just the beginning. This sort of exercises will be continued in this academic institution, and we must, incubate the young minds to develop our country. So thank you all. And so we have a next session, that is our cultural program, and our participants from Asansol Girls College are ready. And with this cultural program, we will end of this today's session. Tomorrow, 11 a.m. sharp, we have a excellent lecture from New Zealand. So, resource person from New Zealand, he will be delivering about the current advances in biosciences. So, it's a request to all of you to please attend the session. And now we have a cultural program under the leadership of Dr. Uttam Mondol, who is our joint secretary. So tomorrow, Ashima Chatterjee, renowned, the botanist, turned biochemist Ashima Chatterjee. Our botany department is named after her. So you can, do visit to our botany department and our experimental setup also you can explore. So tomorrow we have a parallel session of Ashima Chatterjee Memorial Lecture, as well as here some extraordinary lectures from different parts of the country or different parts of the world we are going to witness here in Asansol. So tomorrow will be so exciting, and we are going to get some experiential learning and what we are now and what we are going to become after these 2 days of session conference. You will find a substantial difference. So now I'm going to hand over the mic to our, Honorable Dr. Uttam Mondol sir. He is our joint secretary in cultural committee and a very renowned poet and a professor of Bengali department. He believes in nurturing the young minds through cultural activities. So it's a proud moment to welcome him, sir. Thank you. And now, sir, you proceed with your cultural activities.
Thank you. Hello. আজ সারাদিন ব্যাপী আসানসোল গার্লস কলেজ আয়োজিত ইন্টারন্যাশনাল কনফারেন্সের শেষ লগ্নে আমরা এসে উপস্থিত হয়েছি। উপস্থিত বিভিন্ন শিক্ষা প্রতিষ্ঠান থেকে আসা বিভিন্ন স্টুডেন্টস এবং আমাদের ফ্যাকাল্টি, বিভিন্ন কলেজ থেকে আসা বিভিন্ন সম্মানীয় গুণীজন, সকলকে আসানসোল গার্লস কলেজের পক্ষ থেকে আন্তরিক শ্রদ্ধা ও অভিনন্দন জানাচ্ছি। আমরা গুরুগম্ভীর একটা আলোচনার মধ্যেই ছিলাম সারাদিন, তাই অনুষ্ঠানের শেষ লগ্নে আমরা ছোট্ট একটা সাংস্কৃতিক অনুষ্ঠান খুব স্বল্প সময়ের রাখা হয়েছে, যাতে সকলের মধ্যে একটা সাংস্কৃতিক একটা সম্প্রীতি, একটা সাংস্কৃতিক মেলবন্ধন গড়ে ওঠে। তার জন্য আমরা একটি অনুষ্ঠানের ব্যবস্থা করেছি। অনুষ্ঠানের শুরুতেই আমাদের সমবেত সঙ্গীত পরিবেশন করতে আসছে অস্মিতা, কুমকুম এবং অভনা, ইংরাজি বিভাগের ছাত্রী। তোমরা চলে এসো এবং তোমাদের সমবেত সঙ্গীত পরিবেশন করো।
নিশ্চয়ই তাই জাগে জাগে আমার গান আকাশ ভরা ঘাসে ঘাসে পা ফেলেছি মনের পথে যেতে ফুলের গন্ধে চোখ লেগে উঠেছে মন মেতে ছড়িয়ে আছে আনন্দের নাদ নিশ্চয়ই তাই জাগে জাগে আমার গান গান পেতেছি চোখ মেলেছি ধরার বুকে প্রাণ ঢেলেছি জানার মাঝে অজানার করেছি সন্ধান নিশ্চয়ই তাই জাগে জাগে আমার গান আকাশ ভরা সূর্য তারা বিশ্ব ভরা প্রাণ তাহারি মাঝখানে আমি পেয়েছি আমি পেয়েছি মোর স্থান বিস্ময়ে তাই জাগে জাগে আমার গান আকাশ ভরা
থ্যাংক ইউ। এবার নৃত্য পরিবেশন করবে প্রহেলিকা মাইক্রোবায়োলজি ডিপার্টমেন্ট। আউটস্ট্যান্ডিং পারফরমেন্স আমাদের আসানসোল গার্লস কলেজের গর্ব, আমাদের ছাত্রীরা প্রহেলিকা শুধু আমাদের কলেজের নয়, ন্যাশনাল এবং বাইরে বিভিন্ন রাজ্যেও সে বিভিন্ন জায়গায় তার পারফরমেন্স করে থাকে। এখন আমাদের সঙ্গীত পরিবেশন করতে আসবে অনুস্মিতা। অনুস্মিতা ইকোনমিক্স বিভাগের ছাত্রী, সে অপূর্ব গান গায়, আপনারা শুনলে সেটা বুঝতে পারবেন। অনুস্মিতাকে বলবো তার সঙ্গীত পরিবেশন করতে।
আসিব আসিব এ মাথার ছায়া আবার আসিব ফিরে স্মৃতির তীরে এই বাংলায় হয়তো বা হতাশ হব হাস হব কিশোরীর ভুল প্রতিবেলার পার সারাদিন কেটে যাবে ফুল মিলে অন্ধকারা জলে ভেসে ভেসে আবার আসিব আমি ফিরে আবার আসিব আমি ফিরে বাংলার প্রেমন বিহার ভেসে জলাঙ্গির ভেবে ভেজা বাংলারে এ সবুজ করুণ আবার আসিব ফিরে ধান স্মৃতির তীরে এই বাংলায় হয়তো দেখিবে ছিলে সুদর্শন করিতেছে সন্ধ্যার বাতাসে হয়তো দেখিবে হয়তো শুনিবে লক্ষ্মী পেঁচা ডাকিতেছে শিমুলের ডালে হয়তো শুনিবে হয়তো কোয়েল খান ছড়াতেছে শিশু এক উঠোনের ঘাসে ঘাসে রূপসার খোলা চলে হয়তো কিশোর এক সারা ছেঁড়া বাড়ির দাওয়ায় রাগামি ছন্দাই অন্ধকারে রাগামি ছন্দাই অন্ধকারে দেখিবে অমল হাসিতেছে ধীরে আমারে ভাবে ইহাদের ফিরে এ সবুজ করুণ আবার আসিব ফিরে ধান স্মৃতির তীরে এই বাংলায় হয়তো মানুষ নয় হয়তো সংকোচে শালিকের বেশে আবার আসিব ফিরে ধান স্মৃতির তীরে এই বাংলায় আবার আসিব ফিরে ধান স্মৃতির তীরে
খুব সুন্দর জীবনানন্দ দাশের বিখ্যাত কবিতা তার সুন্দরভাবে অনুস্মিতা আমাদের সামনে পরিবেশন করলো। এবার আমাদের যারা ফ্যাকাল্টি আছেন তারা কোনো অংশগ্রহণ করতে চাইলে অংশগ্রহণ করতে পারেন। বিভিন্ন শিক্ষা প্রতিষ্ঠান থেকে আগত ছাত্রছাত্রী যারা আছো তোমরাও যদি কোনো বিষয়ে অংশগ্রহণ করতে চাও তাহলে অংশগ্রহণ করতে পারো। নইলে আবার আসিব ফিরে এই গান দিয়ে আমাদের অনুষ্ঠানের সমাপ্তি হয়ে যাবে। আমরা আবারও আমাদের বিভিন্ন কনফারেন্স থাকবে, ন্যাশনাল ইন্টারন্যাশনাল বিভিন্ন সেমিনার থাকবে, আবারও বাইরে থেকে সবাই ফিরে ফিরে আমাদের কলেজে আসবেন। সেই জন্য এই গান দিয়েই আমাদের আজকে অনুষ্ঠানের সমাপ্তি ঘোষণা করা হলো। ধন্যবাদ সকলকে, ধন্যবাদ আসানসোল গার্লস কলেজের সমস্ত ফ্যাকাল্টিকে, ধন্যবাদ প্রিন্সিপাল ম্যাম সোয়াতি নন্দী চক্রবর্তী মহাশয়কে। এত সুন্দর একটা প্রোগ্রাম আমাদের ফ্যাকাল্টিরা খুব সুন্দরভাবে সমাধা করেছেন এবং আগামীকালও হবে। সবাই উপস্থিত থাকবেন। আবার আমরা এই আশা রেখে আজকে অনুষ্ঠানের সমাপ্তি ঘোষণা করছি। ধন্যবাদ। আজকে মূল অনুষ্ঠানের সঞ্চালক অর্ণব চ্যাটার্জি মহাশয়কে তাকে অনেক অনেক ধন্যবাদ। খুব সুন্দরভাবে সেই অনুষ্ঠানটাকে এই পর্যন্ত পরিচালনা করে এসছে।